Assalamualaikum. Hello, my fellows. Today we will have a lecture on chemotherapy. So, first we will see the difference between normal cells and cancer cells. Normal cells are small, uniformly shaped nuclei. Have their, this kind of very specific nuclei, relatively large cytoplasmic volume. Now, on the other hand, cancer cells are large. They are very their nuclei shape is not specific and relatively small cytoplasmic volume. Here we see conformity in cell size and shape. Cells arranged into discrete tissues. Here we see variation in cell size and shape, disorganized arrangement of cells. Normal cells we see may possess differentiated cell structures, normal presentation of cell surface markers. Here is loss of normal specialized features, elevated expression of certain markers. Normal cells, lower levels of dividing cells and cell tissues clear, demarcated. Large number of dividing cells, poorly defined tumor boundaries. This is the cell structure. We go next now. We see normal cell and cancer cell. You see, cancer is a disease in which some of the body cell grow uncontrollably and spread to other parts of the body. DNA repair genes are involved in fixing damaged DNA. Cells with mutation in these genes tend to develop additional mutations in the other genes and change in their chromosomes, such as duplications and deletion of chromosome parts together. These mutations may cause the cell's board to become cancerous. Going on next, we again see a major difference. This is the normal cell tumor suppressor gene in normal cell that prevent cancer. And here in cancer cell, DNA changes that inactivate tumor suppressor genes can lead to uncontrolled cell growth. Due to this reason, the cells divide just uncontrollably. The tumor microenvironment, TME, is the environment around a tumor, including the surrounding blood vessels, immune cells, fibroblasts, signaling molecules, and extracellular matrix. The tumor and the surrounding microenvironment are closely related and interact constantly. Tumors can influence the microenvironment by releasing extracellular signals, promoting tumor angiogenesis and inducing peripheral immune tolerance, while the immune cells in the microenvironment can affect the growth and evolution of cancer cells. This is a picture of a tumor. We see that uh, this is the cancer cell and the fibroblast is out of control. Going on next, what is metastasis? Cancer that spreads from where it starts to a distant part of the body is called metastatic cancer. For many types of cancer, it is also called stage 4 cancer. The process by which cancer cells spread to the other parts of the body is called metastasis. Now going on, which many different kinds of chemotherapy or chemo drugs are used to treat cancer, either alone or in combination with other drugs or treatments. These drugs are very different in their chemical composition, what they are made of, how they are prescribed, and given how useful they are in treating certain types of cancer, the side effects they might have. Every time any new cell is formed, it goes through an unusual process to become a fully functioning or mature cell. The process involves a series of phases and is called cell cycle. Chemotherapy drugs target cells at different phases of the cell cycle. Understanding how these drugs work helps doctors predict how drugs are likely to work well together. Doctors can also plan how often doses of each drug should be given based on the timing of the cell phases. Cancer cells tend to form new cells more quickly than normal cells and makes them a better target for chemotherapy drugs. However, chemo drugs can't tell the difference between healthy cells and cancer cells. This means normal cells are damaged along with the cancer cells, and this causes side effects. Each time chemo is given, it means trying to find a balance between killing cancer cells in order to cure control the disease and sparing the normal cells to lessen side effects. The good news is that the most normal cells will recover from the effect of chemo over time, but cancer cells are mutated, not normal cells, and they usually do not recover from effects of chemo. This is why chemo is good at killing many types of cancer cells. Next, class of chemo drugs. Alkylating agents, platinum compounds and nitrosories, antimetabolites, anthracyclines, antimicrotubule agents, vinca, alkaloids, and toxins. Topoisomerase inhibitors, anti-tumor antibiotics, anthracyclines, corticosteroids, other chemotherapy drugs. Now these are the class and examples of and biochemical activity, biological effects of different type of drugs. Anti-metabolites. Examples are 5-fluorouracil as biochemical activities, analog of pyrimidine nucleus. Biological effect is perturbation of RNA and DNA synthesis used in clinical oncology. Colect- Colorectal cancer, pancreatic cancer, non-small cell lung cancer, pancreatic cancer, bladder cancer, and breast cancer. Gemcitabine, same things, and methotrexate inhibits dihydrofolate reduction in folates required for DNA synthesis. Breast, head and neck leukemia, lymphoma, lung osteosarcoma, bladder, and trophoblastic neoplasm. Alkylating agents, cyclophosphamide adds an alkyl group to DNA, inhibition of DNA replication, lymphomas, leukemias, brain tumors. Dacarbazine, metastatic melanoma, Hodgkin's lymphoma. Melphalan, multiple myeloma, ovarian cancer, malignant melanoma, anthracyclines, and these are many more with their uses in clinical oncology and biological effects and biochemical activities. Alkylating agents. Alkylating agents keep the cell from reproducing, making copies of itself by damaging its DNA. These drugs work in all phases of cell cycle and are used to treat many different cancers, including cancers of the lung, breast, and ovary, as well as leukemia, lymphoma, Hodgkin disease, multiple myeloma, and sarcoma. Because these drugs damage DNA, they can affect the cells of the bone marrow, which make new blood cells. In rare cases, this can lead to leukemia. The risk of leukemia from alkylating agents is dose dependent, meaning that the risk is small with lower doses but goes up as total amount of drugs used gets higher. The risk of leukemia after getting alkylating agents is highest about 5 to 10 years after treatment. DNA alkylating medications, anti-cancer agents, this is alkyl group and the other guanine basis and cell cycle. Altritamine, bendamustine, busulfan, carboplatin, carmostine, chlormabuzil, cisplatin, cyclophosphamide. Platinum compounds. Platinum-based anti-neoplastic drugs, informally called platins, are chemotherapeutic agents used to treat cancer. They are coordination complexes of platinum. These drugs are used to treat almost half people receiving chemotherapy for cancer. In this form of chemotherapy, commonly used drugs include cisplatin, oxaliplatin, and carboplatin, but several have been proposed or are under development. Additional of platinum-based chemotherapy drugs to chemoradiation in women with early cervical cancer seems to improve survival and reduce risk of recurrence. Cisplatin. and the trade name is platinol. Injectable solution 1 mg per milliliter. The mechanism of association action of cisplatin has been associated with ability to cross-link with the urine bases on the DNA to form DNA adducts, preventing repair of the DNA, leading to DNA damage, and subsequently induces apoptosis within cells. Administration rate has varied from a 15 to 120 min infusion. 1 mg per minute infusion, 6 to 8 hours infusion, 24 hours infusion, or per protocol. Cancer of cervix, endometrium, prostate, esophagus, kidney, non-small cell lung cancer, head and neck squamous cell cancer, osteogenic sarcoma, bone marrow transplants. 75 to 100 mg scare 4 weeks when used with cyclophosphamide at 100 mg 4 weeks as single agent. Nitrosoureas are a group of alkylating agents that have a special action. The other alkylating agents listed above cannot travel into the brain, but nitrosoureas are able to do so. They can enter the brain because they are able to cross through the area known as blood-brain barrier, a special area that keeps more drugs out of the brain. This action makes these drugs useful in treating certain type of brain tumor. Carmustine, lomustine, streptozotocin. Next indications. BIC and U. Carmustine for injection is indicated as palliative therapy as a single agent or in established combination therapy in the following. Brain tumors, glioblastoma, brain stem glioma, medulloblastoma, astrocytoma. Multiple myeloma in combination with prednisone, relapsed or refractory Hodgkin's lymphoma in combination with other approved drugs. Common side effects of BHCNG include nausea, vomiting, headache, flushing, injection site reaction, pain, swelling, redness of darkened skin color. Metabolites interfere with DNA and RNA by acting as substitute for the normal building block of RNA and DNA. When this happens, the DNA cannot make copies of itself and cell cannot reproduce. They are commonly used to treat leukemias, cancers of breast, ovary, and intestinal tract, as well as other types of cancer. Anthracyclines. Anthracyclines is a class of drugs used in cancer chemotherapy that are extracted from Streptomyces bacterium. These compounds are used to treat many cancers, including leukemia, lymphomas, breast, stomach, uterine, ovarian, bladder cancer, lung cancers. The first anthracycline discovered was donorubicin, which is produced naturally by Streptomyces cereus, a species of Actinobacteria. Clinically, the most important anthracyclines are doxorubicin, donorubicin, epirubicin, and adrabicin. Other types of drugs used to treat cancer. Other drugs and biological treatment are used to treat cancer, but aren't considered chemotherapy. They often have different side effects in chemotherapy. Many are used along with surgery, chemo, and radiation therapy. Targeted therapies. Targeted therapies work by finding specific substance called proteins and receptors that some cancer cells have. The proteins or receptors is precisely targeted by the drug, so normal cells are not affected by the drugs. This is different than how traditional chemotherapy drugs work. Target drugs can be used as the main treatment for cancer, or they may be used after treatment to keep the cancer under control to keep it from coming back. Hormone therapy. Drugs in the category work on the different actions hormones that make some cancers grow. These drugs are used to slow growth on certain breast, prostate, and endometrial uterine cancers, which normally grow in response to natural sex hormones in the body. They work by making the cancer cells unable to use the hormone they need to grow and preventing the body from making the hormone. Immunotherapy. Immunotherapy is a type of treatment that uses drugs to boost or alter a person's immune system. These drugs are used with certain types of cancer to help a patient's immune system recognize and attack cancer cell. Thank you for your attention. We will meet in next topic, inshallah. Allah Hafiz.